Good afternoon, everyone. My name is Chiamaka Agbasionwe, and my research mentor is Dr. Catherine Medler. I work in the Department of Biological Sciences. I'm an intern in CSTEP, and I attend UB. Today I'll be speaking to you about the characterization of a newly identified signaling pathway in taste cells. So, a brief overview of what I'll be discussing today is, first I'll tell you some background on taste transduction pathways, and I'll state my objective, I state my hypothesis, I'll give you a detailed and graphic overview of my methods, and I'll state my results, state my conclusion, future research endeavors, and I'll give acknowledgments to people who have supported me. Okay, so traditionally, they believe that there's only two signaling pathways. The first is ionic, which is dependent on voltage-gated calcium channels that brings in calcium, and the calcium thus releases neurotransmitters to form synapses so that the electrical signals could be sent to the brain. And then the other kind of pathway is complex transduction pathway, which transduces bittersweet and umami. Umami is protein detection, so it's the detection of complex proteins and stuff like that. And basically, to go through this is, first you have a lingered. A lingered is the bitter stimuli, the sweet stimuli, whatever it is. So you take in the food, and then it goes, first the G protein coupled receptors activates PLC beta 2. PLC beta 2 activates IP3R3, which releases calcium from intracellular calcium stores. The calcium thus activates TRPM5. TRPM5 ultimately causes cell depolarization right here and releases neurotransmitters through hemichannels. The most important thing to remember about this pathway is that TRPM5 ultimately releases neurotransmitters through hemichannels and uses ATP. So my objective is to identify proteins involved in the new signaling pathway. The new signaling pathway is unique because it's dependent on G protein coupled receptors, like the complex pathway, but it doesn't release neurotransmitters through hemichannels. It releases neurotransmitters through biochemical stimuli, like you see in the bottom. So it uses components of both the two other transduction pathways. My hypothesis is that TRPM4 is involved in transducing chemical signals in the new pathway. So this is an overview of what's going to be my methods. The two categories of my method is, the first thing I want to try to do is try to amplify cDNA, the cDNA of the mRNA expressed in the tongue. If I could amplify the cDNA, then it's in the brain, and then if it's in the brain, then it's probably in the body. And then the second category of methods is protein detection. So the first method is preparation for immunocytochemistry. Immunocytochemistry is protein detection. It's a way in which we try to detect proteins. So the first thing I do is I obtain a mouse. The two mouse that I'm working with is a black 6 mouse, which I just started working with, and the GAD67 GFP mouse. What makes this mouse special is that it fluoresces green, so it's good for labeling, so I can see the contrast. The next step is that I euthanize the mouse using CO2 in this chamber right here, and this is a CO2 oxygen tank. And then I cut the mouse tongue out and making sure that the CV right here is intact. The reason why we want the CV to be intact is because the CV contains taste buds, and taste buds contains taste cells. The next thing I do, step 4, is I put the tongue in a fix. The reason why I put it in a fix is because I don't want the cells to degrade. Once you cut the tongue out, it starts degrading immediately, so you have to put it in a fix. And then what I do next is I place it in a 20% sucrose solution. I place it in a 20% sucrose solution because I want to prevent it from freezing when I'm cutting. The next step is that I freeze the tissue using OCT compound. An OCT compound is just a tissue that allows it to cut effectively. And I freeze it for 5 minutes. The next step is that I cut the section using a cryostat. I cut the sections in 40 micrometer sections so I could get it thin enough to look at in a microscope. And then I wash the sections using PBS. But mind you, I only collect the sections that have CVs because I want the taste cells. So I wash it, and PBS is kind of like a mouthwash. So now I'm ready for immunocytochemistry. Immunocytochemistry, the first thing I do is I incubate it with block solution. What block solution does is that it blocks out any non-specific binding, so that when I do my binding and labeling, that I'll get my target. The next step is that I incubate the section with diluted primary. The primaries that I'm working with is PGP 9.5 and PLC beta 2. Now, the reason for a primary antibody is because in order to detect the protein, you need a primary that's going to bind to the protein, but you need a specific primary. So these are specific primaries, and then I use that, and then I incubate that for 2 hours at room temperature. So this is kind of, I didn't put no pictures in this slide because it's kind of the same. Then you incubate it and wash it. So so the sections are left in the primary and then washed again the next day. And then after washing, this is a very important step. After washing, I use a secondary antibody. The reason why I use a secondary antibody is because the primary, there's no way to tell that the primary antibody binds to the protein because it doesn't fluoresce, so it's just like we don't know. The way we do know is by using a secondary that's going to bind to the primary. So if the series of binding occurs, then the protein that we're looking for is there. And like I said, these are very specific. So for example, I always use rhodamine GO anti-rabbit because the PGP 9.5 and the PLC beta 2 is raised in rabbit. But the rhodamine anti-rabbit is raised against rabbit. So I have a protein that I'm looking for. I have a primary that's raised in rabbit. And then I have a secondary that's raised against rabbit that's going to bind to whatever that's raised in rabbit. So it's very specific. And then I wash it again and fluoromount it and cover slip it. And then I view the sections using a microscope. So my next method, which is kind of separate from what I'm doing, but it's not like in order, but it gives you understanding. Okay, so reverse transcription polymerase chain reaction, RT-PCR. We all learned in school what transcription is. It's from, you have DNA to DNA to RNA. Reverse transcription is mRNA to cDNA, which is a template. Now, once we have the template, we could amplify the cDNA for our target in the PCR reaction. But mind you, we use specific PCR programs to which proteins that we're looking for. So this schematic right here is for GAD-DH, the GAP-DH program. So if this is amplified, then we know that GAP-DH is in the brain. But if we use, this actually goes to the next thing. So if we use GAP-DH program, we use this GAP-DH to check the quality of our sample before it's used for analysis. But once we use the GAP-DH program, that means that the GAP-DH, and if it's amplified, that means the GAP-DH we're looking, the protein is in there. And then we use TRPM4 to see if the TRPM4 program to see if TRPM4 protein is there as well. So the next thing I do after the RT-PCR is I run a gel. And the reason why I run a gel is to see to separate my PCR products and to see to make sure there's no genomic contamination. And I'll show you an image of that later on. So these are my results. So as you can see, figure 4A is just an image of the taste buds. And then figure B is an image of the expression of GAP-DH 67. figure C is an expression of the rhotamine is an anti-PLC beta 2 labeling. And then figure D is an overlay. As you can see, there's no overlay, which means that it's not co-expressed in the same taste cells, which we expect to happen. But it does kind of, I don't know if you can see the arrows, it does have some overlay, which means that there might be taste cells that have both proteins in it, but generally there's not. And this one is the black 6 mouse. So I used the PGP 9.5 labeling, and as you can see, it labeled very well, which means that PGP 9.5, we could use that to identify cells that have chemical synapses. And we want to identify cells that have chemical synapses because we could identify if the protein that we're looking for in the unique pathway is there. We could use it to identify the proteins we're looking for in the unique pathway. So this is the result of my gel electrophoresis of the GAP DH sample. It worked, and as you can see, the negative control along this line, there's no genomic DNA contamination, and yeah, it worked, and as we expected. So my conclusion is that I'm going to try to use immunocytochemistry and molecular biology to keep on, because my research is not done. I only identified a protein that could be used to label other proteins involved in the new pathway, but it's not done yet. But I'm going to try to identify if TRPM4 is involved in transduction in the newly identified signaling pathway. My future research is to determine if cDNA of the TRPM4 can be amplified, and I'm working on that right now. We're testing it with the PCR TRPM4 PCR program, and we're using different temperatures, adding magnesium, adding less magnesium, just trying to work with it to see if it could be amplified. And then after that, we're going to pursue the specific antibody, primary that can bind to TRPM4, and a secondary as well. So my acknowledgments, these are my acknowledgments. Ms. Owens, I'd like to thank God for giving me this opportunity. CSTEP, Matthew, Tawana, Dr. Flamin for the seminars, Dr. Catherine Miller for being an amazing research mentor who's very strict, but she disciplines me, and my graduate assistant, Michelle. Yeah.
Got 2 questions. You said TRPM4. What is? I saw it's part of the process of finding like certain taste buds or whatever. But what is it like? Do you just label TRPM4? Does it stand for anything, or?
It's just called TRPM4. It's a protein in the TRPM4 channel. Okay. So we're trying to identify if it actually has a TRPM4 channel. So yeah.
And you said you make the slices 40 micrometers thin.
Yeah.
How do you do that? Is it a machine that does that for you?
Yeah, you set it. You put it in the thin and you set it. So every time it cuts, it goes down 40. The cryostat, it goes down 40 micrometers. Yeah.
So what are your next steps?
My next steps is to continue identifying the proteins involved in the new signaling pathway. And right now, I'm just trying to identify, because what we believe I don't know if you remember, but the complex pathway that I showed you had TRPM5. And the new pathway has IP3R3 and IP3R1 and PLC beta 3, which are isomers of the old pathway. So we believe that if it has TRPM5, IP3, and if it has IP3R3 and IP3R1, then maybe also has TRPM4, which is an isomer. So we have two pathways that look like each other, and we're trying to identify. My hypothesis is that maybe the one looks like the other, kind of has the same proteins as the other one has.